How are you? My name is Kohl Perry. I'm a sophomore biology major at Tuskegee University. I had the pleasure of working in the lobby lab over at Olin Hall, and as well as the Joyce Van Eck lab here at Cornell University or BTI. And so this summer, I've had the experience of being able to use and examine lipid nanoparticles as an effective method for DNA delivery in the plants. We've been trying to optimize plasmid DNA cargo delivery into the plant cells of tomato plants, there's a way of transformation already, but using agrobacterium mediated transformation, but it faces many problems such as gene damage, limitation of applicability, and most importantly, low transformation efficiency. So we use this technology, lipid nanoparticle technology, was actually been optimized for therapeutic and nutrient delivery within plant cell, mammalian cells. Most importantly, the Moderna COVID vaccine is an example of this lipid nanoparticle research. So we formulated these lipid nanoparticles, and we had two different ways of testing whether these nanoparticles would be able to transform. Exactly. We had protoplasm infiltration and we had direct foliage infiltration into the plant leaf. And so with the protoplasm, we used a GFP reporter construct, green fluorescence protein, which you can see. This is what the optimal green fluorescent protein will look like. And if you come over here, after we image, this is our positive control of our polyethylene glycol buffer combined with our protoplasm mix after the 2-day step. And we compared this with our lipid nanoparticle formulations to see whether this could work. And in all of our research, we got slight, if you had to look very closely, you had to have a keen keen eye, definitely. So we were able to see a slight, very slight expression, but that's not enough expression to determine whether it's a effective mean of gene delivery in the plants. So in the future, what we want to do to combat this is this formulation space and what they're made of right here. This is what our lipid nanoparticles, the lipid cocktail that encapsulates the DNA that you're trying to deliver, is made of. We use this ionizable lipid right here. This ionizable lipid was responsible for delivering plasmid DNA into cardiomyocytes, which is responsible for the contraction of the heart. Right. So this is one of the 3 ionizable lipids that we use, and we use this. This was also the exact formulation of the mole percentage that we used that was able to deliver the plasmid DNA, which is the cargo that we're working with, into the contraction cells of the heart. So working with this formulation right here in the future will be the most important part to what we can get to optimizing this plasmid delivery into plant cells. So just continue to do more and more leaf infiltrations and more protoplast transformation. I've been telling people all day, if you're in a race, we're kind of like starting line, finish line, be like 2 to 3 strides in. And so we're just going to continue to stride and continue to look for the work and to optimize this plasmid DNA delivery. So thank you.
It's a long race to run in science.
It is.
Are there any methodologies that you had to learn for this that were really fun?
Yeah, this methodology of actually infiltrating the leaf, it mostly it takes a skill to do it. So you kind of have to be very careful with how you infiltrate, and sometimes you'll infiltrate and it won't move. Yeah, you might have a dead leaf. I remember with this ionizable lipid in particular, we were trying to infiltrate this. Well, it was a different trial, but it was like a dead leaf. You could barely get anything into it, and so
between a little bit dehydrated, so it can infiltrate, but it dies. Not ideal.
So that's what I've been working on this summer. Thank you for asking.
Thank you so much for sharing with us. I hope you had a lot of fun with it. It seems like you were busy and you did have a good time. So, well, thank you again.